Welcome everybody to today's seminar. I just wanted to let everybody know how we're going to handle questions and answers. We're going to use the Q&A function of the webinar. And so please just type your questions in there and then Ben can answer them at the end. You can type them as we go if you have, them during the process and then we'll work through that at the end. Also go ahead and feel free to use the chat for things that aren't questions related to the talk. And if you have any questions for me as the host or need anything. Okay, I'm going to turn it over now to David to go ahead and introduce Ben.
Happy Thanksgiving, everyone. Canadian Thanksgiving, that is. On Thanksgiving, I like to spend some time thinking about some of the things I'm thankful for. And on Canadian Thanksgiving, I think especially of good things from Canada that I'm thankful for, like Nanaimo bars and Ben Dalziel. Ben was born near Toronto, Ontario. He did his undergrad and master's at the University of Guelph. And after a couple of years working for the Ontario provincial government in natural resources, he sailed a small boat from Sault Ste. Marie down through Lake Michigan, Lake Erie, and Lake Ontario to Ithaca, New York, where he did his PhD at Cornell. And after a postdoc at Princeton, he joined the faculty here. These days, Ben is perhaps best known for as the primary PI of the team-based rapid assessment of community-level coronavirus epidemics, the TRACE project, for monitoring COVID-19, for which he has worked tirelessly during this pandemic. But his research is much broader than that. In general, he studies population dynamics in social systems, from migrating caribou to diseases such as influenza, Ebola, and coronaviruses. He brings to his work creativity and his deeply thoughtful nature. Ben is now up for promotion to associate professor with tenure, and this talk is one component of that. It gives me great pleasure and great honor to pass the Zoom mic to Ben Dalziel. Ben, take it away.
Thanks, David. Hey, everybody. I can't see you, but I guess I feel your presence, or I have evidence of your presence. Obviously, this isn't the way that we thought we'd be doing this, but I'm happy that everyone's here, and thanks for coming to this talk. I thought I'd start the talk actually the way I start most of my classes, which is at the beginning. And by that I mean at the beginning of the universe. So this is a picture of part of the universe. Actually, it wasn't that long ago, 13.8 billion years or so. Within this century, it's possible that there could be one person on Earth for every year that the universe has existed. Just this one tiny planet in the midst of mostly water, in the midst of billions and trillions of stars and galaxies, one person for every year that the universe has existed, if we get to some of the higher population projections over the next century. So the universe is really not that old in that sense. It's much bigger than it is old. What's even more recent is our understanding of all of this. In the 1970s, when we launched the Voyager probes, which just recently crossed out of our solar system and into interstellar space, in some ways, we're really just at the beginning of understanding the universe and our place in it. On the Voyager probes, there was this golden record because the probes had this goal of maybe one day being encountered by an extraterrestrial civilization or serving as a time capsule for us in the distant future. And they had this golden record on them that contained some information on how to build a record player, a map to Earth, triangulating against neutron stars. And they contained this quote from then President Jimmy Carter that was a greeting to whoever might encounter the capsule. So I think it's it's an interesting tricky problem, right? Like as a president sitting down to write this memo to aliens or to our, ourselves in the deep future. And it's kind of funny to think about how different presidents might have handled that message. But here's what Jimmy Carter said. He said, "This is a present from a small and distant world, a token of our sounds, our science, our images, and our music, our thoughts, and our feelings. We are attempting to survive our time so we may live into yours." And I really feel like President Carter hit the nail on the head, both biologically in terms of the struggle for survival, but also just in this moment, we're trying to survive. So one thing that I think about when I think about the golden record is the Sustainable Development Goals, what we sort of collectively as humans, as as governments, populations, citizens, communities, what we need to do to survive. We need to reduce, eliminate poverty, hunger, improve healthcare, improve equality, clean water. What I'm struck by is how many of these goals require integrating pattern process relationships across scales in science. And more specifically, how many of these goals depend on integrative biology and mathematics. I think that the departments I'm in, integrative biology and mathematics, are actually uniquely positioned to tackle the cross-scale nature of these problems. And I'm delighted to be, in those departments and part of that work, because in part of how it relates to these development goals. So what I see as the central work right now of my research group and my work with collaborators, students, is to understand ecological and evolutionary dynamics in social systems. So what's a social system? Well, sort of a quick definition would be it's a network of relationships that exist between individuals, groups, and institutions. But I actually want to take a kind of a nerdier definition and say, really what a social system is in a statistical, physical sense is it's a system that's far from thermodynamic equilibrium. So, so it's a system that's highly structured, highly non-random, of course. So, and Ilya Prigogine, a Nobel Prize laureate, who studied systems far from equilibrium, actually highlighted this and used a city as an example. He said, are most types of objects around us at thermodynamic equilibrium? Obviously, in a town or living system, we have quite a different type of functional order of special interest to biology. So I'm interested in how ecological and evolutionary dynamics play out in systems like this, these highly structured systems, which are actually quite different than the assumptions of most of ecological theory, which tends to assume that populations are well mixed, that interactions between individuals are essentially random, which is very, very helpful for doing theory. It can perform well in a variety of circumstances, but that assumption is actually exactly wrong for social animals, right? In fact, we have this different circumstances, different nature, and I'm interested in ecological and evolutionary dynamics in that context. And so one way into that, a very specific way into that, is through a phenomenon called superspreading, which is when in epidemics, and it's surreal to present this talk during the pandemic because now there's articles in the Atlantic about superspreading, and so you all already know probably what it is, but just to say, superspreading occurs when a minority of individuals are involved in transmitting the disease to the majority of folks who get it. So it's these bursts of transmission. And these bursts are evidence of underlying structure in the host population, or at least they can be. And so one way into these questions is to think about whether is to go to epidemics where we have a fast system. We have a system where there's both basic research element going on that I really just described, and then also it's a system where the research has the potential to help people, and so it's an attractive system. You go there, and what we're looking for in this way in is super spreading events that can give us an indication of structure, of this social structure at work, altering ecological and evolutionary dynamics. And so I worked with Red Cross just about the time that I arrived at where are we? Oregon State. And to coordinate with them on a program they were doing during the 2013 to 2016 Ebola epidemic on safe and dignified burial. So as you'll remember, one of the key ways that Ebola spreads is through funeral practices that involve touching the deceased. Fluid from a body is incredibly infectious, and so safe and dignified burial was a huge part of how the epidemic was brought under control. And so Red Cross did a lot of that work, and we acquired data on working with them on 200 or so safe and dignified burials of folks who died of Ebola. And by looking at the time that the deaths occurred and where they occurred in space, we were able to use some Bayesian analysis to reconstruct the transmission trees of a plausible reconstruction of whom infected whom. And what we found is that superspreading played a really key role in sustaining the epidemic. So whether you, on the left, we have a map here of the western area of Sierra Leone with the 200 burials that for which we have data. The red dots are what we call index cases. These are ones that were most likely inferred to have arrived from outside the system. The others, the remaining ones are infections that descended from those index cases. And whether you look across the whole system here on the right, looking at the upper bar, or you look at just the secondary cases, you see that a significant quantity of the infections that occurred emerged from superspreading events. So in a random well-mixed system, the kind that typical ecological models assume, this would not be the case. But here we have this highly structured bursty transmission that is at least symptomatic of the impacts of social structure. And we find that it's actually predictable. So you might expect random variation in how many secondary infections are associated with a given case. It doesn't necessarily mean that you're feeling direct effects of a highly structured social system, but we find that actually it's predictable. And so, for example, the potential to be involved in a superspreading event varies by age, with young folks and older folks being much more likely to be involved in those bursty transmission events. And one explanation for that is that these are folks who, during the course of their illness, might be more connected to others through the process of receiving or giving care. We also know that the type of contact that occurs, the type of behavioral interaction that occurs between susceptible and infected individuals, plays a huge role in determining how much infection risk there is, and can, depending on what behaviors occur, generate a burst. So caring for, as I just mentioned, or having fluid contact with infected individuals has a huge impact on the odds of onward transmission that can lead to these transmission bursts. And now this isn't surprising, but what I want to sort of lay out here is, first of all, how much the epidemic was driven by superspreading events, and secondly, the predictableness of it, which really suggests that we're watching a process unfold in a highly structured system. And we can do that with data, with the types of data we collect on epidemics now. That's increasingly possible to actually sense these, so that we don't have to rely necessarily on the well-mixed or mass action assumptions. We also observe structure in how superspreading changed over time. So on the upper graph here, there's the epidemic curve. It's color-coded into different sections. In the middle graph, we're looking at the mean offspring, which is a way of saying the mean number of secondary cases caused by a case over these 5 different time periods. And dotted lines at 1, we need at least each case to cause, well, so we prefer actually it didn't, but so when each case causes on average 1 secondary case, right, that sustains the epidemic. And you can see that, shortly after the peak, right, the average number of cases caused by a secondary, the average number of secondary cases caused by a case was well below 1. And what sustained the epidemic over the following, year were these dots, right, these super spreading events. But at the same time, you can see that going to the lower graph here, the super spreading parameter K was actually changing over time systematically. This graph doesn't show the error bars, but the error bars are small. So confusingly, the super spreading parameter K gets smaller when there's more super spreading. So when you see this line go down over these periods, that's super spreading playing a disproportionately more and more important role in sustaining the epidemic as it goes on. So we have systematic variation, super spreading over time, predictability in terms of demography and behavior, and super spreading playing a key role in driving the epidemic, which is all consistent with this idea of epidemics unfolding in these highly structured social systems, and that having an impact on how they unfold. Because there was this strong asymmetry where most cases were involved in super spreading events, that also means that most secondary infections, in most cases the number of secondary infections was really low. Most transmission bursts, right, are really self-limiting. You have these bursts followed by these rapid fade outs. And that's actually kind of a puzzle. So let's take a little math interlude for a second, and different folks will engage with this at different levels. That's totally okay. You don't need to understand the slide to understand the talk. But I want to take a second here and talk about branching processes. And so here we have an equation for a branching process where Z of T is the number of infections at generation T, ZT plus 1, number of infections in the next generation. Y is the number of secondary infections caused by a particular individual. So if we want to know how many infections there are in the next generation, ZT plus 1, we add up all those Ys across all the different individuals, their contribution in terms of contributing to cases in the next generation. So we sum all of the secondary infections caused from the first case to the ZTth, the last number in that generation, and we get ZT plus 1, and you get this beautiful recursive formula for a branching process. Not beautiful in practice when it's applied to an epidemic, but beautiful mathematically. So Y sub I, the number of secondary infections, the individual reproductive number, we say, of a person, how many secondary cases emanate result from contact with them. We think of that as a random variable. It has this mean, which you could call R naught, which you're all really familiar with now. That's the mean number of secondary infections caused by an infectious individual. And we recover this, this rule that pervades a lot of ecological and epidemiological theory. When R naught is less than 1, when the number of secondary infections caused by a case is on average less than 1, the outbreak will almost surely go extinct. It's not replacing itself. It's somewhat analogous to if the birth rate in a country was less than 1. So, on the other hand, when R naught is greater than 1, an epidemic is possible. This is still a random variable, it's still stochastic. So it's not certain that an epidemic will happen, but it's possible. Whereas if R naught is less than 1, the epidemic is doomed. Okay, so what's the problem? Well, the problem is that when you have an epidemic that persists for a long time, like years, with a mix of these bursts and these fade outs, well, what's R naught? To get the bursts, you need R naught above 1. You need a big R naught to get those bursts. But to get the fade outs that happen the rest of the time, the majority of the time, you actually need an R naught less than 1. And so we have inside this when you take the branching process and you look at the data, there's this mismatch that maybe is a way further into thinking about the impact of social systems, the role of social systems in ecological, epidemiological dynamics. Okay, so let's move systems now and talk about canine influenza. So I'm talking here about avian-derived canine influenza for which there was an epidemic in the States. The data I'm going to show here is from March 2015 up to March 2 years later. It was collected through a collaboration, a network of collaborators, myself, Cornell Veterinary Diagnostic Laboratory, and a range of collaborators that collated these data. And I want to show you how the epidemic plays out because it's another example of this phenomenon that's sort of puzzling when you drill into the theory of these bursts and then these fade outs. Okay, so here's a movie we're watching. Yeah, maybe I should stop for a second and say. So on the horizontal axis we have time. On the vertical axis we have actually, you can't see them, but zip codes across the US from the east to the west. So the orange colors here are infections that are picked up by our surveillance system. The blue ones are tests that were negative, so you can get a sense of what testing coverage was. And we're just going to watch this play out through time and play out on the map. And you can see the whole time series above. So it starts off in Chicago, shows up in Atlanta, shows up in New England, there's cases in Florida, LA. But notice whereas Chicago, there was a massive outbreak that sustained over time. And a lot of the other places, there were a few cases and then it just faded out. And then things got quiet, but the epidemic was still there. It was smoldering along in the Chicago area where it started. So what is R naught in Chicago? Because if it was bigger than 1, you'd get a big explosion like this. So what was that? And why did it happen 2 years later, right? We have this pattern that is actually really difficult to explain using standard theoretical tools from epidemiology, from quantitative epidemiology. Well, we get a partial explanation when we sequence the virus. And what we found is that the outbreaks and the burstiness was sustained from multiple reintroductions from the reservoir. So we have distinct clades happening for the Midwest 2015, Midwest 2016, and then what happened in the Southeast in 2017. And those result from reintroductions, importations of cases. And I think results that are coming in out of the TRACE project now with the genomics we're doing there are consistent with this idea that, whereas when we run epidemic models, we tend to sort of seed them with a few individual cases and then think of the rest of it as happening endogenously. In reality, at least in some cases, we have importations kind of raining down on us, and some of them take off and some of them don't. And this sort of high migration rate of cases in and the relative importance of exogenous forcing, I think is something I think about when we see these data. But it does not provide a complete solution, because even then, how do we explain this long sort of tail in Chicago? What's R naught in Chicago? Here, if you estimated R naught, you'd get something like 2. Here, maybe right around 1, but even so, it would be very unusual for it to last this long. A critical branching process can last for a while, but this is a long time. And if this is happening all over nature, are all the branching processes right at their critical point? And if so, how did that happen? It's either a unsatisfying solution because it's not generic, or it's pointing towards something kind of interesting. Okay, so let's go back to our branching process model and see what we might be able to do with it to account for these dynamics that we're seeing. Well, let's start by thinking about not just the individual reproductive number of a person, but or a host, but the individual reproductive number of the person that they, or the host that they acquired the infection from. So YI is our focal case, and XI is the individual reproductive number of the person that they acquired it from. Now, we already said that these are independent and identically distributed in the standard branching process. So you could sort of say, what's the point? Well, let's write a model anyway. And let's say that we're gonna put these in a log scale, and we're gonna say, let's consider the conditional expectation of what an individual reproductive number should be based on what its parent is. So, and we write an expression for that, and we add a superspreading parameter so we can have variance. And so the reason that we're doing this, and I'll go over in a second, but the reason that we're doing this is we want to capture the idea that if there was systematic variation in structure in the host population, one of the ways that it might come out is by autocorrelation from one individual to the next down the chains of transmission in how infectious or how transmissible their infection was. So you can imagine these pockets of high density, like animal shelters, for example, where it's not just the potential for one isolated superspreading event, but actually you have systematically higher transmission in a whole sort of context there. And so that would, in the transmission tree, lead to these correlations between the individual reproductive numbers that are something that's not in the typical branching process. And so this is the work of a grad student in my lab, Noah Silva de Leonardi, who found a way to sort of tweak the basic branching process model to introduce correlation between the infectiousness of successive infections with this one autocorrelation parameter delta, which provides a way of, in a single parameter, summing up how structured the population is with respect to transmission rates. So the tendency for high transmission rates to be chained together in particular contexts. And so it's a branching process model where the individual reproductive number follows an Ornstein-Uhlenbeck process, for those of you who work on those things. And now we can go back and ask the question, does this rule still hold? This problem, this dichotomy of R naught greater than 1 or less than 1 outbreak, determining whether or not an outbreak happens with this modification. And excitingly, it doesn't. The evidence to date from Noah's work suggests that you can have an R naught of 0.8 for which an epidemic should not occur. That you should have outbreaks die out almost surely. And if you increase the autocorrelation, the delta parameter, the thing that's representing the social structure in the host population, you can actually go from having outbreaks that look like they're subcritical, like they should be fading out, to outbreaks where when they take off, they really take off. And so these are massive outbreaks with R naught of 0.8 by taking into account the structure of the host population. So I'm going to leave this thread for a second and move to the next one, which is to say that none of this is happening in the context of just one microparasite alone. We live obviously in a polymicrobial world, to say the least. And one of the things that the lab's been thinking about over the last few years is about that microbial context in which these dynamics are occurring, instead of just one pathogen, instead of just one microbe, a whole microbiome, for example, a whole collection of pathogens. And so we've collaborated with Oregon Humane Society in Portland to assemble a network of animal shelters up and down the West Coast, and to use the coordinated and highly tracked movement of dogs among these shelters as a way to explore how complex host populations curate their microbial communities and the consequences of that for disease and the way that disease affects that. And so we're thinking of the dog noses with their microbes as kind of these fast-moving islands from island biogeography, and this possible role of transmission there, and how the microbiomes are playing out. And so we're examining the signal of that. And this started a few years ago. An undergraduate in my lab, Anastasia Premilena, who's now an intern at PNNL, assembled this data set that's really cool, I think, of 154 shelters up and down the West Coast. She called all of them, she interviewed all of them. And from the respondents, she asked them, how big are you? How many dogs do you have right now? When you ship dogs out, where do you send them to? And when you receive dogs, where do you receive them from? And she was able to build this network showing these coordinated transfer of dogs among shelters. And you can't see direction on this network, unfortunately, but the red arrows are showing shipment up from California to shelters, primarily to Oregon Humane Society. The blue arrows are showing redistribution out. And so we have this sort of this system that's mixing in a very organized way that will come back later in the talk. It's sort of this highly efficient circulation system that you can imagine having an effect on the way that microbes were being transported around and shared in this highly structured host population. So a graduate student, Liz Knorr, in my lab dug into what the microbial consequences of that would be by swabbing many dogs at Oregon Humane Society. Liz lived up there for a while and just swabbed dogs on intake, and then every day thereafter until they got adopted out, looking at how the microbiome changed over time, what pathogens they had, what their disease state was. And one of the first things we noticed was how common co-infection was. So many dogs, whether symptomatic or not, were carrying multiple respiratory pathogens. And you can see that here, we're sorting by the number of samples, the number of pathogens present, and looking at the number of co-infections, and half of them are showing multiple pathogens there, whether symptomatic or not. And some shedding at very high levels. This is inverse to the CT score. So Liz also did 16S work to look at the microbiomes of these dog noses, and so this is a metacoder plot that actually shows sort of a bacterial tree of life, and it's colored by how different, it's comparing sick and healthy dogs and looking at differences in their microbiome. So when you see orange, it means something, it means that bacterial taxa is overrepresented in sick animals, and when you see green, it means it's overrepresented in healthy animals. And you can see there are these, it's preliminary results, but there are these slight but significant differences between sick and healthy animals in terms of what their microbiomes look like. Interestingly, the presence of respiratory symptoms was also associated with an overabundance of obligate anaerobes. By monitoring the microbiomes over time, from the moment that the dogs arrived at the shelter, Liz was able to track how the microbiomes changed over time to see if arriving at a new shelter, arriving at a big hub, could actually, what the effect would be on the microbiome. And the preliminary results there are that while dogs arrived with significant differences depending on where they came from in terms of their nasal microbiome, by just 3 days later in the shelter, the microbiomes were much less dissimilar, and they all resembled the sort of what may be the sort of classic Oregon Humane Society microbiome. So there was this rapid apparent change in the microbiomes of the dogs over time, concurrent with the microbiome being involved in mediating sick versus healthy, either cause or consequence. We're not sure. So conceptually similar work is part of an NSF EEID grant that I'm a co-PI on. Anna Jolis is the PI, and there's a number of other co-PIs. And the goal here is to really understand these relationships between the microbiome, pathogens, selection, both on pathogens and host. And then of course the immune system is lurking in the background of all of this and needs more consideration and doesn't get considered enough because it's hard in my field. So we're doing this big field study on desert bighorn sheep, where we have different metapopulation structures across different populations, and we're able to hopefully titrate out some of these dynamics. And Isaac Julio, a recently arrived PhD student in my lab, will be working on that. All right, so I often think of these sort of social structures that we are using as proxies for social structures generally. I often think of them as also stand-ins for human cities. I mean, cities are where an increasing majority of humanity lives. They are these incredibly structured aspects of our sort of human life history, I guess you could say. And so for me, thinking about epidemic dynamics in cities and ecological dynamics in cities is fascinating, and forgive me, but I think it's important. I mean, urbanization and global change are two of the main sort of things that are going to shape life for us over the next 100 years. So, so I want to talk now about disease dynamics in cities, in human cities. And to do that, I'm going to use a data set from electronic medical records that looked at 3-digit zip codes across the US. That's equivalent to about the size of the city. For insurance claims, for insurance billing codes that reflected influenza-like illness. And so by using these data, we're able to get a sense of the incidence of influenza-like illness at the zip code scale, 3-digit zip code scale, on a weekly basis across the US. And this is a resolution of data that hasn't typically been available. And so even though, flu spreads at the scale of meters and days, minutes and days, our data that we typically have for flu is much coarser than that. So this is an exciting opportunity to dig in and see, one of the predictions you could make is if ecological and epidemiological dynamics are playing out differently because of social systems, then maybe cities that look different should have different epidemic dynamics. And that's something we've never been able to test for flu. This is a slide that says that the data is good and it works. So what do we get? So if you plot, this is the plot of across 600 zip codes of their adjusted proportion influenza-like illness, which is just influenza-like illness adjusted for reporting. And the thickness is because we actually have 600 lines here, one for each zip code. And so what do we see in this thing? Well, first of all, you see, different patterns in different years, and that's due to the evolution of the virus. As we know, some years are bad flu years. The virus makes an antigenic shift to something that the proportionally less people are immune to, and we see a spike in influenza cases. So you can kind of roughly see the signal of that as the difference between a peak one year and a peak the next. So when A. Fujian 411 showed up in the 03-04 season, we had this bad flu season spike. You can see the differences in the spikes there. We also see the impact of climate variation. So influenza in the temperate regions is sensitive to climate. We'll get into that in a second. And so if you look at the distance between sort of like the middle of the gray caterpillar thing and the troughs and in the peaks, you're seeing climate modulation affecting flu seasons happening in the winter. But there's a third sort of piece here, which is the thickness of the caterpillar, right? This is the width of this thing is the variation at a particular point among those 600 different cities, unexplained spatial variability in epidemic dynamics. So the null hypothesis, and it's quite a lot, right? It's comparable in magnitude to what you see from an antigenic shift or from climate variation. It's massive. So the null hypothesis here is that, there's randomness, there's measurement error, different cities go different ways at different times. One year, one city zigs and the next one zags, and that creates variability. The alternative, though, is that some cities are systematically different than others year after year. And that's what we find. So using a measure called epidemic intensity that I'll tell you about in a second, what we found is that some cities are systematically more or less intense in their flu epidemics year after year after year. So here in the blue, I've outlined within that gray caterpillar the 10 least intense cities where the incidence in flu is really, really spread out. It's not focused in a particular time. It's spread out throughout the season more. And it's the same 10 cities, right? Here we have the 10 most intense cities where where the incidence is proportionally focused way more just around flu season. And it's the same cities year after year. So why would that be? Well, to dig into it, let's quantify it. So here we have a not very intense city where incidence is spread out, and a pretty intense city where it's all focused together. So we can measure intensity as the inverse of Shannon entropy. So when we have everything focused down into one place, that's like having one species in your pool, and the Shannon diversity is very low. So we take the inverse of that, we get a very high intensity. So we're using inverse of Shannon entropy to measure intensity. So now that we have that, we can do that for a particular city, and we can map it. It's just a statistic for each city J. So we map it, and we see there are these striking patterns where in the east we have all the intense epidemics, and the coastal metropolises tend to be less intense. We also see the signal of climate, specifically specific humidity, affecting intensity. So places with high seasonal swings in specific humidity have intense epidemics, and that's actually to be expected. We know that specific humidity modulates transmission. When the air gets really, really dry, the respiratory droplets get really small. Ballistics, they travel further. The virus is also more viable in temperate regions when the air is dry. So we get the risk posed, the sort of cloud of risk around an infected individual expands out when the specific humidity drops. Not the relative humidity, but the specific humidity, the proportion of mass, the mass to mass ratio of water in the air. So what was more surprising, though, is that epidemic intensity was really related to population size. So small places had really intense epidemics, and the big, highly organized places had less intense epidemics. Now, why would that be? Now, this was really surprising to me because, in fact, if anything, I would have thought that the big, highly organized, and I have to be honest, like this is kind of what I was angling for, I guess. It was my pet alternate hypothesis. We're not supposed to have those, but the big, highly organized cities that look like viral bioreactors, right? That they were going to have these massive, intense epidemics, and that the small places would be more random. And here we're seeing the opposite. So why is that? Well, this is just to say what I just said, which is that, as cities get larger, they get more highly organized. And this actually does turn out to be important. So here's a picture really quickly from some work from before I came to OSU, drawing out the commuting patterns in Canadian cities of different sizes. And as they get larger, we have more and more highly organized commuting patterns where disproportionately more people from around the city are are funneling in during the day to these few focal work locations. It's highly far from thermodynamic equilibrium. Okay, and you can see that here too, that, you know, as sort of the daytime population where people are when they're at work, as the city size gets big, that the crowding in those locations, it goes up disproportionately. And big cities are more highly organized for their size during the day. So residential organization scales with population size too, but not as fast as work population size does. So the movement of people in cities is disproportionately more organized in big cities, and it leads to these intensely organized patterns. Okay, but as we said, I would have thought that would have led to more intense epidemics. So what is going on? Well, to understand what's going on, we have to take a look at how flu spreads. And so, as I mentioned, the proportion of the virus that stays viable in the air, it's something we can measure experimentally in different environmental conditions. You sort of spray a bunch of virus into a drum with a particular specific humidity, come back a certain amount of time later, see how much of the virus is still viable. So if we sort of impose some sort of arbitrary epidemic threshold where we say, like, you know, on average, given how close people get together, you know, you need 50, let's just say 50% of the virus needs to stay viable for R naught to be above 1, say, or whatever it needs to be above to get an epidemic. So now we have a range of the season, right, when we can have a flu season. So the thing is that the minutes that elapse between shedding and uptake, the minutes that determine, of course, how much virus we have when we come back. So we set the specific humidity and we walk away. If we walk away for 5 minutes, and when we come back, there's going to be more viable virus than if we walked away for 30 minutes, right? So what if we thought of that as a proxy for contact patterns? So what if in large cities, people are more closely drawn together in a more organized way, and the average time between shedding and uptake decreases? One way sort of story to think about this is, yes, it matters what the specific humidity is for the transmission of influenza, but it matters a whole lot less what the specific humidity is if you're sitting right next to someone on the subway. So these slides are sort of explaining that. So to understand how that leads to less intense epidemics in metropolises, we need to put this into a model that thinks about susceptibility. And the punchline really is that if you think about large cities as being able to, the trick is to think about large cities as being able to transmit influenza efficiently over a wider range of climatic conditions. What this graph is saying is that it matters less in big cities what the climate is. The big cities sort of are able to incubate. And what that means is more early season transmission, more late season transmission. Because flu is an immunizing infection, though, that early transmission in the metropolises is immunizing. And so you actually get more and more transmission over a wider period of the flu season, which leads to the incidence being more spread out. Conversely, or on the other hand, in small cities, there's a relatively narrow range of climatic conditions under which the flu can spread. And so susceptibility isn't depleted by early season transmission the way it is in metropolises. Instead, the susceptibility builds up until climate conditions allow an epidemic, and then it all happens at once. So that's the hypothesis. We make the hypothesis into a model. We simulate the model and we see something that looks reasonably like the data. And in fact, if you parameterize the model with demographics from each of the cities and run it forward, you see a striking resemblance between the model and the data. The features of cities in terms of their climate, their population size, their level of crowding and organization, and their movement patterns, you put all that in, you can really closely match the epidemic dynamics that you're observing over really long time scales. So it suggests that it's evidence in favor of this hypothesis, and also it suggests the potential for long-term predictability in the flu, which hasn't been something that has really seemed possible. It suggests the potential for that based on fundamentals of the characteristics of a city, the climate, their structure. So that has recently led to a collaboration, sponsored research with a company, Kinsa, that has a network of smart thermometers that monitor, anonymously monitor body temperature in cities across the US. And this, whereas before we were working with historical data, in partnership with Kinsa, what's exciting is the potential to do this in real time, to actually generate real-time, long-lead local influenza forecasts. And those have recently also been applied to COVID, to detecting emerging COVID hotspots in a way that's a leading indicator that precedes what you'd see from case data from the formal healthcare system. Wow, time is really flying. So I have one more thing to talk about. 1.1 more things to talk about in the next 10 minutes or so. And what I want to do is pivot from talking about epidemics, in case anyone's tired of hearing about epidemics. And I want to talk about how this fits into ecological theory. And the bridge here is that, unfortunately for us, an epidemic is, and especially a cyclic one like this is a consumer resource cycle with us as the fuel, us as the resource. But it's a consumer resource cycle nonetheless, and it's this really important object in ecology for understanding how ecosystems work, the consequences of trophic dynamics. And there's consumer resource cycles like this all over the place. A classic one, of course, is the snowshoe hare lynx cycle. They're really, the tools that we use to understand them quantitatively are really similar. So when we're talking about the impact of social structures on epidemic dynamics, on the patterns in epidemic cycles, we're really talking about how social dynamics, how the collective behavior of animals and social systems affects consumer resource dynamics, and that has ecosystem implications. I think I'm going to skip. Ahead a little bit. Well, no, this is worth saying. No, I'm going to skip. Okay, so what we want to do is to start, we're going to do a theoretical ecology exercise, and we're going to start with a classic model of a consumer resource system, the Rosenzweig-MacArthur model. And we're going to try to add in collective behavior. We're going to try to add in individual behavioral interactions scaling up to create behaviors at the population level that you would not see in independent individuals. And that can be seen in everything from migrating caribou to the structure and movement of cities, the building up of societies. So we're going to try to add that. And we're going to use collective movement, actually, sort of flocking, to represent collective behavior, because it's a relatively well-studied type of collective behavior. So we start with this consumer resource model. We have two differential equations, one for the resource, one for the consumer. N is the population size of the resource, P is the population size of the consumer. The first term here is just standard density-dependent logistic growth of the carrying capacity K in the resource. Then we have consumption. This fraction here is the functional response, right? So this is the per capita rate of consumption of the predator, of the consumer, based on the resource density and on how long it takes them to handle each resource item and their encounter rate with the resource, their per capita encounter rate, which is sometimes called the search rate. Now that feeding term here appears down here, but multiplied by B, this is turning resources into offspring, and then MP is a mortality rate in the consumer. So here we go. So this model has some wonderful features to it that let us think about ecosystem stability and coexistence. And one of them is the paradox of enrichment. It's this counterintuitive phenomenon where if you increase the carrying capacity of the resource, you go from a place where this model has a stable equilibrium, resource and consumer sort of flatlining here, and we increase the carrying capacity and we increase it, and we move into these cycles of consumer resource dynamics, which get progressively up more and more likely to have the system go to extinction. They go to extinction right when they hit the zero here. So why would increasing the carrying capacity of resources, enriching the system, you'd think that that'd be a good thing, sort of like dumping in fertilizer or something, but it leads to this instability. And it leads to a big question, which is like, how can an ecosystem be stable when, in the face of, if something like this is so prevalent and can destabilize it? So the paradox of enrichment is one of the ways that we ask the question, how can ecosystems be stable? And that question endures. Okay, so let's see what collective behavior does to this situation. And the way that we're going to do that is we're going to implement this model as an individual-based simulation. Here's a little picture of the arena. This is an oversized picture of one individual and their horizon of interaction with other individuals, so it's local. And we're able to deduce what the encounter rate should be, the fraction of the arena that they're able to see, the rate at which they encounter other individuals. So we can implement this model using the same parameters here and have the model behavior match what these parameters say, as long as there isn't a bug in the code. And then, because it's an individual-based simulation, we can turn on collective behavior and see what happens. So that's what we do. So here we have 10 replicates. That's why there's 10 turquoise lines for the resource and 10 turquoise purple lines for the consumer. And we're watching up to time 1500, we're watching consumer resource cycles, just like they should be. Loca Volterra, Rosenzweig MacArthur, there's the links in the hair, or influenza. And then we turn on collective behavior, and all of a sudden, consumer resource cycles are gone. They just flattened out. So we go from this place where, and here's the analytical prediction in black and the simulations and the thin lines. We go from this place where we're seeing just what we thought, which is these cycles, to collective behavior causing a spiraling into this equilibrium. There's another way of looking at that. You can look at a bifurcation diagram where we go from a steady state to oscillations right at the theoretical point when we predict that we should. In independent individuals, the simulations and dots match the lines of the analytical predictions. When we turn on collective behavior, something happens. The stability conditions appear to have changed, and in fact, the system can persist under levels of enrichment that would drive independent individuals extinct. Something that I'm definitely not going to have a time to talk about today is that in addition to apparently resolving, at least in this instance, the paradox of enrichment, this addition of collective behavior also resolves the paradox of the plankton, which you may know about. It's the question of how many similar species can persist on a few number of resources, like plankton in the ocean, where there's just relatively few dimensions to their niche. And so what we see here is that actually an inferior competitor that just sort of less efficiently handles food, just basically has a lower capture rate, drops it more often, that competitor should be eliminated rapidly by competitive exclusion in the case of independence, but with collectives, it persists indefinitely. So this is something that's at the core really of ecological and evolutionary theory, and when you add collective behavior to a canonical ecological model, it changes. So what's going on? Well, the first hint of what's going on is if you look at really high carrying capacities, what you see is that you get, even though with collective behavior turned on, you still get these things that look like population cycles. And when you plot them, in phase space, they look like population cycles. But what's weird here is that there seems to be an additional state variable. So here, if you look at the shadow, don't look at this thing yet, look at the shadow of this thing on the consumer resource plane. Here are our consumer resource cycles. But we've added a third state variable, the encounter rate, the per capita encounter rate, which is supposed to be constant. It's the probability in an instant of time that a randomly selected consumer and a randomly selected resource will be within capture radius of each other. It's not supposed to vary. And you can see in the independent case in the inset here that it doesn't. But when we turn on collective behavior, it does. And it varies in this really striking way, where as resources get scarce, the encounter rate goes down, which actually protects the system from the resources being consumed right into extinction. And the system has this resilience somehow emergent when you add these social dynamics as collective behavior. And after a lot of work, I and colleagues, this is work in collaboration with James Watson and CEOs Mark Novak and IB and Steve Elner at Cornell, I and colleagues have identified that what we believe is happening is that there's a social ecological feedback loop that I'm only, I think, going to have time to go through really briefly. But on one hand, what we see is a fluctuation, underlying this fluctuation in encounter rate is a fluctuation in the number of consumer groups. And so when the number of when the consumer population forms into just a few groups, what emerges is a little society where there are haves and have-nots, and the individuals that are eating have systematically higher exposure to resources than the individuals who are questing, because these groups have a leading edge where they have access to resources and an internal part where they don't. So a hallmark of a social system is systematic variability and access to resources that emerges under a simple model of collective behavior. It affects encounter rate, modulates ecosystem dynamics in a way that promotes stability. So when there's a lot of groups, so in the limit, when every individual is its own group, right, we see encounter rate go back up to the theoretical prediction that it should be. So that's the social affecting the ecological. On the other hand, we have the ecological affecting the social. In environments that are highly enriched, we believe that that enrichment leads singleton groups to be more likely to recruit another individual before they go extinct. And for groups that are growing really fast, because there's a lot of resources, they're more likely to undergo fission. And so that together means that as resources grow, the number of groups grow. And so together, this feedback loop means that resources and encounter rate, consumption rate are being modulated together in this emergent way. Really fast, we can approximate that with an analytical function. When we plug it into the model, what we see is that it actually seems to recover the discrepancy between what the analytical model said and what the collective behavior simulations were doing. Remember, the analytical model didn't have collective behavior in it. That was the point. So we add this correction for collective behavior, and we can recapture what the dynamics should be. So now we have an ODE model that has an approximation of collective behavior, variable encounter rate built into it. I presented this at the Math Bio seminar in the math department here, and Enrique Ton and Patrick Deliner and Jan Medlock jumped on it, and together, and with them doing the mathematical work really, we were able to show, at least for this approximation of the impact of collective behavior, why that's happening. And what we found is that, first of all, under collective behavior, when you increase carrying capacity, which is normally destabilizing, the shapes of these milk lines are really different, and they include this refuge in phase space for where under low resources, you're guaranteed to get resource growth. Whereas if you crash the resource population in standard Rosenzweig-McArthur, you can end up in a place where resources are still being depleted by the predator, even though they're really low. But here, there's this sort of buffer, and that's not guaranteed to lead to a change, but it's sort of an intuition into it. The other thing that they found that I'm especially excited about is the fact that if you enrich a system that has collective behavior, and then you to the point where it's unstable, it turns out that if you increase collective behavior further, you can stabilize it again. In fact, for every enrichment that might destabilize the system, there's a compensatory increase in the strength of collective behavior that will stabilize it again. And I think that I am going to talk for 10 seconds about trace, and then I know that we are out of time, and I would like to save the no time that we have left for questions. Hopefully folks can say I apologize for going late here. So about April of this past year, and then the pandemic happened. And since then, I have been involved in this wonderful, crazy project, leading it, team-based rapid assessment of community-level coronavirus epidemics. We have done door-to-door surveys and wastewater monitoring in 4 communities. We've encountered a wide range of prevalences. We're still early on in analyzing what all the data means. I'm really happy that in some cases, I believe that the information that we were able to provide helped communities make decisions that allowed them to navigate changing conditions of the virus. I want to highlight that this trace in its very early days in terms of my journey with it began because of a really innovative collaboration that a grad student in my lab, Rachel Aber, set up with Student Health Services, where we were collecting de-identified nasal swabs from folks, or diverted medical waste from folks who were coming in for respiratory pathogen tests. And because those swabs were already in the freezer, and because I knew from Liz's work that we could get virus out of nose samples, I started thinking about the untapped capacity of university labs to do this type of work. I thought, gosh, if the theoretical ecologist's lab has the capacity to run plates and plates of these types of samples, couldn't there be something we could do? And that was the beginning, and it really existed because of work that grad students were doing in my lab. And so I want to acknowledge them, and also the wonderful PI team and OSU-wide effort that's made TRACE possible. With that, I'm going to close and just say some very quick thank yous to the funders, the collaborators, to my departments, IB and Math, to OSU, all across OSU, the many, many offices and individuals who've supported the work, especially to the College of Science, to my lab and Mark Novak's lab, to my mentors who have been very generous, my family, and to my partner, Jessica Bliss, who is the fiercest, most wonderful collaborator, and none of this would have been possible without an extraordinary amount of support from her. And with that, I'll thank you all for listening, and I hope that there's a little bit of time left for questions. I'll turn it over to David. Or do I just read through them?
You can just read through them. Thank you, Ben.
Okay. So Dave Lytle says, nice seminar, Ben. Given your results about collective behavior and the paradox of the plankton, how do your findings affect the relevance of Peter Chesson's modern coexistence theory? Specifically, the rather strict assumptions about intra versus inter-specific competition needed to achieve coexistence. That's an awesome question. We've been working to try to fit our results into the MCT. No, it's an awesome question. Also, I have a slide for it, so I'm especially, let me see if I can get, so I didn't get a chance to talk much about the coexistence aspect, but I want to just say real quick that we think that the same mechanism, social ecological feedback loop that we described in the talk also affects coexistence by creating, by releasing species from competition when they become rare. So by making their growth rates positive as they become rare, which is one of the key parts of the MCT, right? We get coexistence by making growth rates positive when the species become rare. And we think that we see that happening. And so I put up the slide here, but I'd love to talk more about that. Thanks for the question. So Bill Bogley asks, are there dynamic similarities between ecological and political systems? I don't think, I feel like I'll get myself into trouble answering because I tend to be someone who wants to overgeneralize. I don't have results on that, obviously. I think that's a really interesting question. I'm aware of the work of Peter Turchin, who's an ecologist who's turned to using some of those same tools to try to predict the rise and fall of human societies. And I think that work is really interesting, and that, of course, why wouldn't those tools be applicable? So yeah, I think that's a great question. Thank you.
Any more?
Any more questions?
And it looks like there's one in the chat that got entered into that chat function from Thomas Schmidt.
In the chat.
I can read it for you too, if that would help. Just let me know.
Oh, I got it. Thanks. One needs only systems of only 2 ODEs for almost all of this, question mark? You speak of R naught being greater than or less than 1, but wrote equality in one case. Please say more about R naught equals 1. So 2 questions there. Sure, okay. I think that the direction we're heading with the super simple approximation for collective behavior in the ODEs is actually toward a system of 3 differential equations. So more broadly, right, I think we want to describe the behavior of this system in terms of its macroscopic state variables. That's the sort of dream. So if we want to add a differential equation, maybe we'd add one for the number of groups. What I think is the case, and what some subsequent experiments, simulations have revealed, is that there's a fast-slow thing going on. So the equilibrium group size, if there were a third equation for the number of groups, that would equilibrate relatively quickly. And so it collapses back down to 2, and we have and we try for an analytical expression for that equilibrium group size, plugging back into the 2. But that's a great question. I mean, I think that's open. That's open. In terms of R naught, please say no more about R naught equals 1, the critical case. Can you say more about your question? Sorry, I want to make sure I get the right angle. I'm not sure if you can just talk. Or...
I can now, yes. I was done.
Oh, great.
Yeah, sure. The mathematics indicates a trichotomy. You indicated that it was a dichotomy, and you put 1 with one of the 2 cases. Why make that choice? What about this boundary case? Does it ever occur? Is it uninteresting for some reason?
I think I'm guilty of dismissing it because R naught equals exactly 1 is the chances are vanishingly small in nature, but maybe I'm not thinking about it right. How would you? This may just be a defect in how we're thinking about it. Tom, without your facial expression, I'm not sure if it's hard to read. Should we, maybe we should talk.
Just sure, let's go to the next question, no problem.
Okay, I look forward to talking about that with you. Yeah, thanks.
Other than the very important question from Jessica about when you might come home for dinner, are there any more questions? Well, if not, then please join me in thanking Ben for his very interesting talk.
Thank you all.
Thank you.